Hello students, welcome you all on my channel. In my previous video you had studied about the various type of nitrogenous waste materials related to the chapter excretory system. In this video I am going to discuss about the excretory system of human. The organs I'm going to discuss, the basic organ, the kidney. Here I will make a realistic diagram to demonstrate the internal parts of the kidney. Along with kidney there are other organs which play crucial role in elimination of nitrogenous waste material through urine. So the number one is kidney. It is pair. Number two is ureters pair. Number three is urinary bladder. And number fourth is urethra. These are the tubes which store or which take the urine from the kidney and helps in elimination in the form of urine. This urinary bladder act as a temporary reservoir that I will discuss later, and about urethra also and ureter also in the separate heading. First I am going to make a realistic diagram, this time not schematic. I will make a realistic diagram to demonstrate the internal structure of kidney, and then you will come to know about the each and every structure clearly. So I'm going to erase this. So I'm going to make you the LS, longitudinal section of human kidney. Oh, it is so. This will help you to understand properly. This structure I am making, this is the pelvis region. This is the pelvis region. Blood vessel entering into the kidney. Because blood is required for filtration. Here the blood is coming from the renal artery, and again the blood will be taken back by the renal vein. So this region is the pelvis. This region is pelvis, or simply it is called renal pelvis. Renal pelvis. This structure, this vessel is a renal vein. This vessel is a renal artery. This structure is the calyces that I'm going to draw. This structure is called calyces. It is of two types, major and minor calyces. This structure is called hilus. This is the hilus, depression of the kidney. Now I'm going to make the structure of the medulla region of the kidney. It is striated. It is a striated region, pyramidal striated region. This is the pyramidal. Numerous pyramidal regions are there in the kidney. I will also discuss briefly These structures, what are the role, why they are in striated in appearance, what is the reason behind its striation. There are numerous, it is, kidney is laden with numerous renal pyramids, pyramidal structure. The kidney has a rough appearance, and this rough appearance outer side is known as the cortex. Kidney is divided internally into two regions. The regions are the cortex and medulla region. Cortex and medulla region. The cortex is granular, that's why I am making dot like a structure in this diagram to demonstrate you why it is granular. I will also tell you about why the structure appear as a granular and why the structure of the medulla is striated. I had told you this structure where the medulla are the fixed, this pyramidal. See, medulla has two parts. The broader end lies outside towards the cortex region. The broader end, and the conical structure is fitted to the calyces. It may be major or the minor calyces. The conical structure towards the pelvis region, So this is the structure. Now kidney has a tough fibrous structure called, I'm going to make an outline structure here to demonstrate you. This is the fibrous structure, act as a protective agent. This is the tough fibrous structure which covers the kidney, The granular structure, the dotted granular structure is called cortex region, cortex of the kidney. It is granular. And this structure, the depression, the cortex region has invagination towards the pelvis region. These invaginations are there. This is called column of Bertini. Remember this invaginated structure. This invaginated structure is called column of Bertini, and this is extending towards the pelvis region. This structure, it is a renal pyramid, and this region is called medulla region of the kidney. Medulla region of the kidney. This is LS of human kidney. LS means longitudinal section of human kidney. Listen that you will find every structure same which is present in human in other mammals. Same structure you will find. See, I'm going to tell you one thing. This renal vein is entering into the kidney and containing a less amount of urea. Always remember, renal vein contain less amount of urea, but the hepatic vein contain highest concentration of urea, because urea is synthesized in the liver, and in the renal vein urea is very less because the renal vein is taking and the filtration of blood is taking place. So obviously the quantity or the concentration of urea will be less, because the maximum urea is filtered by kidney and it is sent through ureter along with urine. So I hope this structure is clear. Now I'm going to give you a lecture on the kidney. See, so you may draw this structure. I'm going to erase this to demonstrate, to give you lecture. See, our kidney is a pair of metanephric kidney. First, kidney, the first point. A pair of metanephric kidney, a pair of metanephric kidney. As you know, I told you about the pair of metanephric. Metanephric means it is posterior kidney, it is highly advanced kidney, and it is also known as posterior kidney. Why it is known as posterior kidney, that it will be clear. Second point, a pair of metanephric kidney lies at the posterior side of dorsal aorta. Dorsal aorta running from the posterior region, and then there you will find the kidney. That's why it is the posterior kidney. It is called posterior kidney. It is lies outside the coelom in abdominal region. In the abdominal region, I'm telling, it lies in the abdominal region. It lies in the abdominal region and outside the coelom. Each kidney lies in the posterior region of dorsal aorta. Dorsal aorta. The third point is, the kidney is covered by, ventrally it is covered by peritoneum. It is a membrane which covers the kidney. Kidney has depression called hilus or hilum. It is a bean-shaped structure. It has depression. It is called hilus. Kidney possesses depression called hilus. In human, the right kidney is slightly lower than the left kidney. Why? Because it has to accommodate large liver. But in the case of a rabbit, just opposite. In human, because we are studying the human, a right kidney is slightly lower than a left kidney, because it has to accommodate a liver. Internally, kidney has two parts. Internally, kidney has two parts: cortex and medulla. And medulla. See, the cortex region of the kidney is cortex is granular in structure, and medulla is light and striated in appearance, because cortex is richly packed with cortex is packed with Malpighian corpuscle. Malpighian corpuscles. What is Malpighian corpuscle? Malpighian corpuscle is glomeruli and Bowman's capsule, or simply it is called glomerulus and Bowman capsule, collectively called Malpighian corpuscles, and PCT and DCT, proximal convoluted tubule and distal convoluted tubules, are present in the cortex region. That's why the cortex region of the kidney appears darker and granular in appearance. Now the medulla region is lighter and striated in appearance because it contains many conical structure called renal pyramids. It is striated because the medulla region of the kidney contains a loop of Henle of juxtamedullary nephrons. Through hilus, pelvis, renal pelvis, and the renal pelvis, ureters leave, while blood vessels like renal vein and renal artery enters along with lymphatic vessels. So this is about the cortex region, and this is about the it's about the hilus region. So I told you the medulla region of the kidney is lighter in appearance because of the nephron. Kidney contains two types of nephron. One is cortical nephron, another is a juxtamedullary nephron. What are the difference between them? The juxtamedullary nephron have larger loop of Henle, and the cortical nephron possesses shorter loop of Henle, that I will discuss in my next video when I will make the realistic diagram, and I will explain which region have the cortical and which region have the juxtamedullary nephrons. So I'm going to erase now. Students, see, kidney possesses, remember this, possesses two types of nephrons. And as you know, nephrons are a structural and functional unit of kidney. It is also known as uriniferous tubules. It is also known as uriniferous tubules. Two types of nephron, namely cortical nephron and the second one is juxtamedullary nephron. Juxtamedullary nephrons. It is present in the cortex region, cortical nephron. Juxtamedullary, it is present in the cortex as well as the medulla region of the kidney. Each kidney is packed with 1,000,000 nephrons. 1,000,000 nephrons. So this is all about the kidney, and I am going to make one flow chart to demonstrate the structure of nephrons, which part of nephron play what role in the kidney for elimination or reabsorption or ultrafiltration. Nephron. As you know, nephron is a structural and functional unit of kidney. Nephron is composed of, it is composed of two parts. Nephron is composed of two parts, consists of two parts. The first part is called Malpasian corpuscles. It has two parts. Glomerulus and Bowman's capsule. Bowman was scientist. The main function of glomerulus and Bowman's capsule is it mechanically filters blood. It mechanically filters blood. Now come to the other part, and this part is called tubules. It is further it is made up of three parts, comprise of three parts. The first one is PCT. It is called PCT. Everybody is knowing the full form of PCT. It is proximal convoluted tubule lying close to the Bowman capsule. Convolution is there. The second one is a loop of Henle. Or Henle's loop. And the third one is DCT. The main function of PCT, it reabsorbs, focus here, it reabsorbs 75% of water, potassium, or collectively I am writing salts. Glucose and amino acids. Loop of Henle act as counter current. Loop of Henle, it act as a counter current and maintains concentration gradient. I am writing here because space is not here. So loop of Henle, it is the first point. Second point I am writing here, it is the loop of Henle. It maintains concentration gradient of urine. That is to say, the concentration of urine depends upon the length of loop of Henle. Larger the loop of Henle, larger the concentration of urine. For instance, in kangaroo rat and in camel, the loop of Henle is very large. That's why the urine of camels are syrupy. And in the kangaroo rat, the urine of kangaroo rats is always semi-solid. It is exceptional case in mammal. Now DCT, it it act as a tubular secretion of H ions, certain drugs, potassium ion, and Na ion, sodium ion. Drugs means there are certain immune suppurative drugs are there, and without the action of DCT it is not possible to eliminate those drugs like methotrexate, cyclosporine. So students, it's all about the human excretory system related to the kidney and the nephron. I hope you have understood these structures, means the realistic diagram and this flow chart of the nephron. So in next video, I'm going to make realistic diagram of the nephrons, cortical nephron and juxtamedullary nephron, and I will discuss its functions there. Thank you very much for watching my video. Stay tuned, more to come. Please don't forget to subscribe my channel, like and share. Thank you very much.